Among 200-odd competitors in the race to create an effective vaccine against the SARS-CoV-2 coronavirus, mRNA vaccines have emerged as the surprise frontrunners. Two candidates, from tiny American firm Moderna and pharmaceutical giant Pfizer, teamed up with German partner BioNTech, claim to boast about 95% effectiveness in protecting people from catching COVID-19. The active agent in both candidate vaccines is messenger RNA, or mRNA. That begs the question, what seems to make RNA so good at promoting immunity? The impressive figures from mRNA vaccines were surprising because initial media coverage suggested that a more high-profile candidate like the so-called Oxford vaccine, ChAd-Ox1nCoV19, or officially, AZD1222, developed by AstraZeneca and Oxford University, would be first to cross the finish line. Instead, it was Pfizer and Moderna who announced that their drugs had almost completed Phase 3 clinical trials, which test safety and efficacy in thousands of participants. Vaccine candidates are grouped into 6 main types based on the technology, or platform, used to expose your immune system to viral molecules, enabling it to recognize and target a virus when your body's defenses encounters that virus. 5 of those 6 technologies are focused on exposing the body to proteins, either on the surface of a virus, vector, vehicle, or as protein subunits. The viral proteins serve as antigens, molecules that prompt your immune system to generate antibodies. After exposure to an antigen, the adaptive immune system learns to recognize, and then target, that specific antigen. For SARS-CoV-2, a vaccine's target is the spike protein that enables the virus to invade a cell. Here's what makes an mRNA vaccine different. Rather than exposing your defenses to a protein directly, the antigen is delivered indirectly via a very roundabout route. The mRNA molecule carries the genetic instructions for producing a spike protein and, once inside a cell, that spike gene is read by cellular machinery to make spike proteins. It's a weird way to expose an antigen, but it works. Essentially, the body receives a target antigen, such as the coronavirus spike protein, despite the fact that an mRNA vaccine doesn't itself contain the antigen. Most cells continuously collect and present the molecules within themselves on their outer membrane. They do that to prove to the immune system's sentinels, such as T cells, that they aren't infected by a virus, as the proteins that a virus produces while replicating should appear on an infected cell's membrane. Sentinels use surface receptors to check the proteins on a potentially infected cell. That antigen-presenting cell, APC, isn't invaded by a real virus if you receive an mRNA vaccine. RNA molecules are transferred, so it's technically not infected, but transfected, with a gene that encodes the spike antigen.